Thank you so much, Dan. That was great. In the interest of time, we're going to move through the presentations, and then we're going to have the questions at the end for the panel discussion. So next, I'm delighted to introduce Iain Gardiner from Simcyp Certara. He's going to share with us some of the envisioned impact for TB drug development of the physiologically-based pharmacokinetic platform that was developed out of the CPTR modeling and simulation team under his leadership.
So, good morning, everybody. So this talk, again, is going to be split into two parts. In the first part, I'll talk about our approach to physiologically-based PK modeling and show you some applications really in the field of drug interactions and how this technology is being used. And then the second part of the talk, what I'll do then is move on to tell you about the work we've been doing over the last 3 years with CPTR to develop some PBPK modeling approaches that hopefully we can then apply to use in the field of TB drug development. So basically the approach that we use for PBPK modeling is to look at using in vitro in vivo extrapolation together with physiologically based models to try and predict the drug concentration and effect in virtual individuals. And what you can see at the top here is really just a simple example of in vitro in vivo extrapolation. So what you're doing is you're taking information about metabolism of the compound in an in vitro system, using scaling factors to predict what the intrinsic clearance of that compound will be in the whole liver. You can then combine this kind of information with a mathematical model of the human body, and that's shown here in a full body PBPK model, where each of the organs is represented by a physiologically realistic compartment with physiologically realistic size and flow in and out. And then basically the idea is, and using the mathematical modeling, you can predict the drug concentration in the different compartments of the body, and then obviously link that to drug effects. And for instance, in the heart, we have a poster showing how you can link the concentration in the heart and use that to predict some of the cardiotoxicity effects you might see. So these are the kind of things you can do with this PBPK approach. The other thing that we do is a little bit different, is to try and really look not just at a single individual, but try and predict what's going to happen in the population. So what we do is we take all the demographic, biological, physiological information that we can, put all of that together to generate a virtual population that we can then use in these kind of simulations. And what we're doing is really to use a kind of systems pharmacology approach, where we take information that we know about the biological system. So in this case it's the age of the individuals, information about their weight, tissue volume, composition, cardiac output, together with information about the drug data. So here we have information just based on the physicochemical properties, and also information on the in vitro metabolism, permeability, and solubility. You can combine that with information about the dose, administration route, the frequency, and look at what happens in different populations. So combining all of this data together in a in vitro in vivo extrapolation model together with the PBPK, you can then start to predict the absorption, distribution, metabolism, excretion of the compounds in the population of interest. And the reason that we take this systems pharmacology approach and split the data apart is that then you can keep information about the biological system the same, and you can change the drug. So if you switch from one drug to another, what's going to happen in terms of the pharmacokinetics of that compound in the system. The other thing you can do is to keep the drug information constant and then look at what happens in different populations. So here you can take information about the healthy volunteer population, and then you can change the information that you have. So you can go to look at what happens in a cirrhosis population, in an obesity population, or in populations of different ethnicity or different age. So this kind of systems approach is really good for looking at how the drug's going to behave in lots of different scenarios. So just a quick refresher of why the ADME properties are going to be important for drugs. So really the absorption, the distribution, metabolism, and excretion of the compound is really going to control the concentration of the drug that's in the blood, and ultimately the concentration that's going to reach the bacteria in the lung granuloma if we're talking about TB treatment. So really understanding all of these processes and how the drug's getting from the blood to the bacteria is really important if we want to get successful therapy. In terms of metabolism, the main enzymes that we need to consider, or the main enzymes I'll be talking about today, are the cytochrome P450 enzymes, or the CYP enzymes. I'll talk a little bit about an acetyltransferase because, again, they're important in the metabolism of some of the compounds we've been working with. And in terms of excretion, I'll just briefly mention some of the drug transporters, in this case, P-glycoprotein or PGP. So the CYP enzymes metabolize a lot of drugs. Of these, the most important ones is CYP3A subfamily. And these are really important because they metabolize 50% of marketed drugs, and they're also subject to drug interactions. So this is one of the areas where we've been working extensively with the PBPK models, and I'll show you some of the results in a minute. But the thing to remember with the CYP3A enzymes is that they metabolize a lot of drugs. They can be inhibited when you give more than one medication together. This can lead to increases in the drug concentration for certain victim drugs, and also they can be induced by a lot of compounds. So obviously rifampicin is widely used for the TB treatment, and it induces a lot of enzymes, including the CYP3A subfamily. And therefore, when you give rifampicin together with other CYP3A substrates, the concentrations of those drugs may be much lower, and therefore it's important to know this kind of information. So this is showing prospective drug-drug interaction predictions using the physiologically-based pharmacokinetic technology that we've developed. And this is a study that was done both prospectively and independently from ourselves. You can see on the top row, what we have here is a simulated AUC ratio from the PBPK model plotted against the observed AUC ratio on the x-axis. And you can see there's a really nice correlation between what was simulated and what was actually observed when the clinical study was finally done. If you look on the right-hand top panel, you can see the simulated over observed AUC ratio just plotted as a ratio. Ideally, what you'd like to see here is all of the data lying on this line of 1. You can see, again, it's pretty good. There's one study that's slightly outside. But in terms of predicting drug interactions through inhibition, this was really quite successful study. On the bottom panel, again, it's just showing a similar kind of data set, but for induction. And again, there is a pretty good correlation between the simulated and the predicted, between the predicted and the observed AUC ratios. So again, this is just showing the application of the technology in terms of predicting drug interactions. And this kind of approach now is being used quite routinely in submissions to the FDA. So this is data that was updated in 2014. But over the past, say, 5 years, there have been 136 different submissions to the FDA using this kind of PBPK approach. And you can see the different areas where the PBPK approaches are being used. So in this case, it's mainly around drug interaction, but you can also see there's a fair number of studies where they've been looking at application of the PBPK approach to predict drug concentrations in pediatric subjects, and then a range of other studies as well. As well as being used in just in submissions to the FDA, there's now a growing impact of PBPK approaches on drug labels. So over the last 3 years, there are 19 examples where actually, if you look at the labeling for any of these drugs, then there's overt mention on the label of PBPK simulations and how they were used in reaching some of the conclusions about the behavior of the drug. And what I'll do in the next couple of slides is just to show you one example. So the example I'm going to show you is this one here. So this is ibrutinib, which was an anti-cancer compound. So the key things about ibrutinib is it's a CYP3A substrate, as I was talking about previously. It's high clearance and has high susceptibility to drug interactions. Now, the sponsors had already done 2 clinical studies. So they'd done one clinical study with a strong inducer of CYP3A, and one clinical study with a strong inhibitor of CYP3A. So what was then done was a PBPK model was constructed, and you can see that the results from the PBPK model actually mirrored really well what was observed with the strong inhibitor and the strong inducer. Once the model then was verified, what they did was to use the model to predict a number of scenarios with moderate 3A inhibitors, moderate 3A and weak 3A inducers. And all of these studies were purely done by simulation, so there was no clinical verification for this part of the study. When you look at the wording in the label then, what it says for ibrutinib with 3A inhibitors is that simulations using PBPK modeling suggest that for a moderate 3A inhibitor, then you're going to get perhaps a 6 to 9-fold increase in the AUC, and for a moderate 3A inducer, a 3-fold decrease in the AUC. They also went on a bit further, so rather than just using that wording in the labeling, what they also did was to take the simulations and then to use the PBPK model to provide some information on dose optimization. So here we're looking at the moderate CYP3A inhibitors. And the recommendation was that if you're going to dose this compound with a moderate CYP3A inhibitor, it lists a number of the CYP3A inhibitors here, then you need to reduce the dose to 140 milligrams. So typically you're giving a dose of 560, you drop in the dose 4-fold to 140. And the reason for this was that they knew there was a fairly flat relationship with safety and with efficacy across this dose range, and they had linear pharmacokinetics between 140 and 1400 milligrams. So the basic idea was if you drop the dose to 140 milligrams, in the presence of the CYP3A inhibitor, then you're going to see a sort of 5 to 9-fold increase in AUC, which would still be in the window that they deemed to be safe, and therefore, based on the modeling, they could just make this recommendation of the dose that they could use with a moderate CYP3A inhibitor. So that was just trying to show you the approach we use for PBPK modeling and show you some of the uses that this type of approach is where it's been used in regulatory submissions. What I want to do in the last part of the talk is to tell you about what we've been doing in collaboration with CPTR over the last sort of few years. So in terms of the PBPK modeling that we've been working on for TB drugs, there are really 3 components. So we've been working on a lung model and also being able to predict distribution into granulomas. I'll show you some data on the virtual South African population that we've been working on, and also we've developed a number of compound files for some of the drugs that are commonly used in TB therapy. And I guess some of the idea of this, some of the applications that you can do with this type of modeling would be to predict first-in-human trials, optimization of dose selection, and certainly the quantification of drug-drug interaction potential. So talking first about the PBPK model. So I already mentioned this briefly, but really what we're talking about is using a mathematical representation of the human body. And you can see each of the compartments is defined, and you have the flow of the blood from the arterial to the venous and then back through the lungs. Obviously, in terms of the TB drugs, what we're interested in is really having a good understanding or a good model of what's happening in the lung. So if we zoom in to look at what's happening within the lung, we created a multi-compartment model where each of the lobes is represented, so the 5 different lobes. And we can model the movement of the drug from the blood into the tissue mass and into the epithelial lining fluid as well. So this is a PBPK model for the lung. Obviously, the other thing that we're interested in is being able to predict the drug distribution into the granuloma. So we've extended the lung model to also include a model that accounts for granuloma formation with the different compartments, and therefore you can start to model movement of the drug from the blood into macrophages, and from the macrophage in the rim into caseum of the granuloma. So this model now is developed, and we've been doing some testing, validation of the model. In terms of the population, we've created a virtual South African population again, so that you can kind of simulate the clinical trials in populations where they're being tested. And so in creating the virtual South African population, what we were looking at was using real data to describe the age distribution and the age, height, and height-weight relationship. So all of that information is included in the model. From the height and weight, obviously, we can describe the body surface area, hematocrit, the protein concentrations. And obviously, we need to account for genetic differences in different populations. So in particular, in the South African population, there's a high incidence of CYP3A5 extensive metabolizers that we need to account for. So the extent of extensive metabolizers is about 80% in this population, would be about 20% in a North European population. And we can also account using the systems pharmacology approach for the physiology changes that occur in TB. So we know that weight and BMI, for instance, are lower. We know that alpha-1-acylglycoprotein levels go up and the albumin levels go down. We know that the hematocrit is lower, and also that the lung pH has changed. So all this physiology information that's available, we know is different in TB subjects, could affect the drug distribution into the different compartments. We can take account of in the model. So this is really what we've been working on. In terms of the compound files, I'll just show you one example. So this is the moxifloxacin compound file, and you can see the predicted and observed concentrations are reasonably well described. So this is just looking at the plasma concentration. We also looked at predicting the concentration in lung tissue and in the epithelial lining fluid for moxifloxacin. And again, we could see reasonably good relationship between the observed and the predicted data in this case. The one thing that we did have to account for with the moxifloxacin model is we had to account for the action of transporter, PGP, in movement of the drug from the lung mass into the epithelial lining fluid. And that's just shown on the next slide. I'm not sure how well this will come out, but it's just showing that to accurately capture the epithelial lining fluid to plasma ratio, then we needed to account for the effect of PGP. So there's 2 slides left. So this one is just really talking about PBPK simulations and how we think they can help. So here you can move from plasma to predict the concentration in the granuloma macrophages or in different regions of the granuloma, and you can link these to pharmacodynamic pharmacodynamic effects. Obviously, some of the questions that you could potentially use this technology to answer then is, what happens if you're giving your medication with DDI and it's altering the concentrations? What's the impact going to be? You know, what effect will food have? What happens in different subjects with different ages, different disease states? So all of those questions are amenable to answering using this kind of PBPK approach. So what I tried to show is this technology is now routinely used to predict drug interaction in the clinic. It's used in submissions and is also being used on drug labels. We've spent effort over the last 3 years to develop the models that you need to do the same thing for predicting lung disposition for the TB drugs, and we're now at a point where we need to show the utility and applicability of those approaches. And the last thing is a quick plug for the workshop that we're planning to run hopefully sometime in August. Thank you.